Thank you very much, and I'm really excited to have a talk here, and I really enjoy the spirit of this conference. So thank you very much for the organization. Yeah, so I'm coming from the German Center for Neurodegenerative Diseases. We are located in Bonn, Germany, and they are running a facility which is called the Laboratory Automation Technologies, where we can, in principle, we are combining drug discovery and high-count analysis screening as well, but also we are established now in the last years an iPSC technology platform. So we provide also like external users, like drug discovery companies, but also internal research groups with this kind of technology platform to conduct drug discovery projects, or either when there's a need of like upscaling iPSC technologies. We have established in the last years the iPSC-derived neuron differentiation, astrocytes, and microglia differentiation, and also the brain endothelia differentiation on the robotics. And we are equipped with 30 plus different working stations or like machines from regular like liquid handlers, dispensers, whatever you need for upscaling your experiments. But also we are running fully automated spinning disk microscopes from our collaboration partners Yokogawa. So to come to the topic, I would like to speak about the human blood-brain barrier and how can we upscale a human blood-brain barrier system to be able to better predict whether a drug goes through the brain or not. So because there's, we have seen in other talks also during this week that there's a bottleneck for drug discovery in general, that only the minority of drugs or novel drugs can enter the brain, and in particular, because I'm coming from the DCNE, from the German Center for Neurodegenerative Diseases, we are in particular interested in generating drugs which could enter the brain finally and like cure dementia or other ZNS diseases. So the human blood-brain barrier, as we have seen a couple of other slides, a couple of different talks this year, it is a specialized endothelium, and it keeps the brain homeostasis. It is required for healthy brain function, and it prevents all the toxic compounds from the blood. They cannot enter the brain because you have the within this brain barrier, you have your efflux transporters. They are like pumping actively out what's coming in the endothelium and push the drugs back into the bloodstream. So that's the general challenge what I have brought across already. In ZNS diseases, there's obviously a need for better predictable models to have like a screening platform in hand for better prediction. So the question arises: how can we do like this, or can we build this human blood-brain barrier in a miniaturized system to screen for permeability? So you might be familiar with all the different blood-brain barrier systems. So in the very schematics here, you have the transfer system. It's a 2D system. It's really nice, and for upscaling, absolutely the system of choice. Then you have the dynamic system, the microfluidics. And you have also the complex 3D systems. So you have here self-organizing tubes. We have seen it in different talks that you put also neurons, you put astrocytes, pericytes all together to have really good physiological properties. And they mimic really nicely the state of the blood-brain barrier in the humans. So the question is now, how can we combine the systems, but how can we, have physiological properties in an accessibility or upscaling for screening system? We have been started with also the iPSC-derived endothelial cell differentiation. We choose this IPS differentiation because they provide so far the best properties to build a blood-brain barrier. We have established the differentiation process from IPS to brain microvascular endothelial cells, which is published by the Schuster lab, and we have put this in our differentiation automated workflow to generate our cells. So one thing I would like to mention is we are using for the maturation step conditioned media. In this particular cases, we are using human brain vascular pericyte conditioned media. When you think about having an upscale system, you don't want to have parallelized differentiation process of your astrocytes, neurons, pericytes, also during a screenable run or during a screening run. Therefore, we did a lot of tests to get similar properties of our blood-brain barrier system with conditioned medias. The advantage of these conditioned medias is you can prepare them in bulk, we freeze them, and then we thaw them when we are doing a screening. So this is what we get. What we got as a starting point when we have when we put these cells into the into the 2D culture. We have here like benchmark properties. We generate. We have done first of all the characterization of this BMEX. They express all these endothelial markers. They have tight junctions. They have other transporters, and also the efflux transporters are present, PGP and BCRP, for example, shown here. And the nice thing is that they also have a really high and physiological relevant tear value, which can be like compared with rodent models. So we reach around 3,000 ohm per square centimeter here, which is like a benchmark thing. And by applying now, I don't show the data here, by applying now the conditioned media from the pericytes, we can even go higher. So let me then go further. We are then going into the 3D model. So we chose the mimetas plates because just simplify, it's just simply because it is a standardized 384 well plate formats. We can use all our liquid handler machines to change media and also to seed the cells or seed the extracellular matrix inside the middle lane here. We have the suitability for imaging. It's beautiful for upscaling because every microvessel inside looks or is the same because it has the same size for kind of a robustness, and it is a perfused model in 3D. So by the gravity flow. And I don't want to go into much detail because I guess you are familiar with the system. We are just harvesting the cells from 6 wells, then we seed it in a top channel, and we let the cells sit, and then we apply the perfusion. After 2 days of culturing under perfusion, you have a fully matured microvessel formed. So all the markers I've shown you before, they are also present in the microvascular tubes. So you have again the endothelial marker, you have the tight junctions, and also the efflux transporters as well. And what we have done is then we further characterized how does it look in 3D when we applied like 3D microscopy by using our spinning disk systems. Here is the full representation of one of the tubes. This is with a tight junction label. Here you have the ECM gel, so this represents the brain side. Also from there shown, this represents the brain side, this is your blood side. And we applied also this cell mask, for example, to see whether you have small holes inside your microvessels. And what we could see just by characterization is that there's also the possibility that you have wound healing, so the cells are still proliferate, but they keep, and this is really important, they keep a monolayer of cells. So I will, hopefully that works. So this is the same representation now, and we did some analysis in Imaris, and here you see the full tube inside, the blood side, and on the other hand, your brain side. And this is just to show you how deep we can go, also with the microscope, how good we can do the characterization. Really striking was also when we look at these characterizations more closer, we could see that for the efflux transporter, for example, here for the BCRP, it is you have a polarized localization inside the endothelial cells, meaning that the BCRP is somehow only present at the blood site and not at the brain site. And that was really striking when you do a combined staining with GLUT1, for example. GLUT1 is your glucose transporters. It has to be on the both sides. It's also shown in this paper to really have the sugar transport back and forth. But the BCRP was just on the blood side, watching for toxic substances. So next is we would like to identify our functionality. So we have to do a leak tight test, of course, to make sure that during our screenings we do not have any false positive effects because we have leaky microvessels. We apply rather small fluorescent molecules here as tracers. So we started with 10 kD dextran, which is our larger molecule, and we have also a very small fluorescein, which has the size of a small molecule with 300 Dalton, around 300 Dalton. And what we do is we apply that to the top lane, which you have your microvessel inside, and then you just calculate really easily the intensity ratio of intensity gel divided by intensity top. And when you have values around 0, you have a leak tight scenario, otherwise you have a leaky scenario. That's just a representation how that looks when you look at multiple multiple chips on the plate. And this is our quantification. So here's the dextran, here's the fluorescein, and what you see is that the dextran is really blocked when you have a leak tight microvessel, and the fluorescein is almost blocked. So I don't have here the permeability values for you, and they are still in production, but for the tracers, we are just using them to making sure that our microvessels are leak tight during the full drug exposure, and we have done it for 5 hours, as you have seen here. And making sure that you have, in this time window where you apply your drugs, you have a leak tight scenario. One thing I would like to stress is we also put here like a negative control, so we treat it also with staurosporine. So you see here it pokes really holes inside the microvessel collapse. Yes, thanks. And the whole system pushes then to a really high ratio. So you might ask yourself, how can we do that more quantitatively? Because like identifying leak tight microvessels just by eye is not really working, not really science. we applied a general, regular k-means clustering analysis where we apply 6 different 6 different ratio data sets in principle to identify our leak tightness. And here's just the 2 ratios of the fluorescein and the dextran together. The thing is that you have always a very narrower window when you look at your intensity ratios when you have a leak tight scenario. So we use this kind of variance because to divide between leak tight and leaky vessels, and this can be done on the fly on the microscope. We have done this analysis directly when we applied the drugs, and you can say at every time point that the microvessel is still intact and there's no toxic effect or there's no general break in the microvessel. One thing was also very striking. We have developed our own microvessel tear device. You could also reach values around 1000, and really beautiful was to see that we could also correlate our tear measurements when you have a leak-tight microvessel, that it absolutely negatively correlates with the fluorescein ratio as well. So that means when you have a high tear, you have a lower fluorescein ratio, and on the other hand, when you have a lower tear, meaning it's not really, it's a bit less tight, you have a fluorescein ratio which is a bit higher. So I come then to the last slide. Then I will show you a couple of compounds we have tested with this system. We are still running a lot of more of reference compounds at the moment, but still this is what we have done, and we have applied a functional assay to detect our to detect whether a drug goes through the blood-brain barrier or not by using a PBMC assay. What we are interested in is an anti-inflammatory drug, because it has been shown for neurodegenerative diseases that these drug candidates is like a really interesting group also to treat and to stop the progression of dementia processes. Therefore, we look at anti-inflammatory drugs at the moment, and in these 2 scenarios, you see you apply your compound of interest in the top lane. You have an impermeable drug, for example, here, and we take the supernatant out and apply it to the PBMC assay. And then the PBMCs are activated, so they are primed by LPS and then activated, and then they have a regular IL-1 beta release. So on the other hand, when you have a permeable drug which goes through and you can take the supernatant out from the bottom and you put that on the on the PBMCs, you have a decrease in IL-1 beta release. So we have looked before we have done this assay in those responses that all our drugs respond in such a way, and also to have a good let's say concentration in this lower lane to also detect sensitively the compound which goes through. So this is the quantification of that. This is the control, so regular IL-1 beta release as we have seen it before, and then we have tested here some of the candidates. These guys are known for like caspase inhibitors. They are known to be not permeable for the blood-brain barrier, and this is what we can show as well. We have also here emricazan as a candidate. It's not purposed for the brain at all. And from our in-house screenings, we have also identified 2 really striking compounds together with our collaboration partners. These are also anti-inflammatory drugs, as you can see here, and this goes in a quite of strong permeable way through the blood-brain barrier. So thanks a lot. My time is over, and I would like to say thank you to my group, to Eugenio Fava, Philip Denner, Birgit Kukowski, and our collaboration partners. Thank you very much.
Thank you, Sven. We have time for one question only.
Thank you, great presentation. So I have two questions. So one is, do you envision this as a PD assay, like the PBMC assay, or do you want to actually measure permeability? And also, as someone who's also trying to push this, but from the pharma side, in your opinion, what data do we have to generate to make sure this generate added value than traditional tools like MDCK overexpressing MDR1, PGP?
So, sorry, there's really a bad noise here. Can you just speak a bit louder?
Okay, so first question, do you plan on actually measuring permeability of the molecules? And the second question is, what additional data, in your opinion, we have to generate to convince people to use this instead of the traditional toolkit like MDCK expressing PGP?
Absolutely, yeah, very good point. First of all, we would like to measure the permeability of drugs by using a mass spec readout and not only the functional assay. And to the second point, it is then we need to really look for more reference compounds that are known to be in the blood, which go to the blood-brain barrier of the humans. And also, we would like to combine our assay with in vivo data from mice, for example, to really match like a linear correlation for that. So in terms of we measure the permeability first, and then we can correlate that to in vivo data. That's the next step, of course, you need. Thank you.
Thank you.
Okay, I think it's fine. Thank you very much.